So again, now at this point, we've treated well over 70 dogs with canine osteosarcoma. To date, knock on wood, we have not had an in-field recurrence, meaning that we've not had tumor develop back in the field that has received the high dose of radiation therapy. And so this is very promising. We verify that with histology or looking at tissue, and to date, we're still batting 1,000, which is tremendous. Typically, we're treating on 3 separate days, so we're giving 12 gray. That's our unit of measure for radiation, in 3 separate fractions. So we're getting a cumulative dose of about 36 gray into the tumor. And our median survival time to date has been equivalent to amputation and chemotherapy or limb spare and chemotherapy. And in dogs, the median survival is about 1 year when we combine those modalities. So very comparable to our other modalities. This form of limb sparing obviously has benefit if the animal has other concurrent neurologic disease or other concurrent orthopedic disease that would make them a less than ideal candidate for amputation. And you can see here we have a Rottweiler that is in the immobilization device on our couch in the Linac, and we're basically immobilizing with non-invasive plastic here below the lesion. So you can see here very big firm swelling in the radius consistent with where the location of the tumor. So we're immobilizing in front of and behind the tumor so that we know each day that the tumor is where we want it to be. And then we verify that with x-rays. And so you can see we have 2 sets of x-rays. We have one from the plan that shows us where basically the blueprint, if you will, of what we need to be to replicate the plan as we've made it, and then that day's position. And then we can actually overlap those and make sure that the bones are lining exactly, and that allows us to ensure that we're treating the tumor with high doses and still avoiding the surrounding normal tissues like other normal bone and skin. And to date, for the most part, our complications have been well tolerated. In cases where we do have very lytic bone, the complications have been predictable and been such that we've been able to intervene with prophylactic surgical stabilization. So going in dogs that we know are at a higher risk for fracture because their tumor is already at an advanced state, and plating those and stabilizing those such that the animal will still be able to benefit from the limb and benefit from the procedure. And now I'm going to hand it over to Dr. Ryan, who's going to talk a little bit about how we're going to take our experience from dogs thus far and use that and apply that in a rat model to answer some questions that we still haven't answered with our dog model and how that may be applicable to humans as well. So, Dr. Ryan?
Good, thank you. So I think this is a really good example of where we can take a condition that's present both in people as well as in dogs, and we can try evaluate a new therapy. So the same as sort of phase 1 clinical trials, that's really the equivalent of what we've been doing with the radiation therapy here in dogs, and it's helped and it's shown a lot of promise. But often when you get something that's promising, it ends up raising a lot of new questions. Radiation can actually be detrimental in the longer term, so we don't know what the long-term effects of radiation could be. So before we go jumping into people, we need to know a little bit more about that. Our survival times, as Dr. Custer said, in dogs is relatively short, so we don't really get to see late-term effects of radiation, but that could be a really big issue for translating this to children. Yep.
I had a quick question about dose. 36 gray sounds like a horse's dose to me. I mean, that sounds like a huge dose of radiation. Is it a lot? Did you use more?
Yeah,
purpose? I mean, what's...
No, so great question. So there's 2 different ways to, and chime in if I can speak. There's sort of basically 2 broad ways you can deliver radiation. So the first is traditional fractionated radiation therapy, and that's where you give a small dose of radiation at each treatment fraction, and you give that over a longer period of time. So that's the Monday through Friday deal, generally for 3 to 5 weeks, for a total. If we were treating most tumors, we want to get to 54 gray, sometimes a little bit more, but that's generally what it takes to get adequate local tumor control. And it's always a balance between killing the tumor and the radiation side effects. The difference with the stereotactic radiotherapy is that we can, because of the image guidance and the way those multi-leaf collimators work, we can actually get a much larger dose per fraction. So instead of giving 2.5 or 3 gray, we can give 12 gray in one dose and still not have the detrimental effects. If we tried just doing it with the parallel opposed, we're going to get bad side effects to skin or nerve or muscle in that area. The radiobiology, so the way the tissues, both the tumor as well as the normal tissues, behave with large dose per fraction radiation is very different to the way it behaves with small dose or normal fractionated sort of schemes. And so osteosarcoma in particular is traditionally been a very radio-resistant tumor. So we've done studies before where we've given traditional fractionated radiation therapy and haven't got very good local tumor control, have had unacceptable side effects, and so because of the way osteosarcoma behaves, it doesn't respond well to normal traditional fractionation. And so that's why the stereotactic radiotherapy with its large dose per fraction, we've actually shown that we can kill it at what is actually a lower total dose because we're giving large dose per fraction. Not bad for a surgeon, eh?
Very good.
So, but the question is, is 36 as low as we can go? We have treated some dogs with either a single fraction of 30. We've done a couple of dogs where, through circumstances we could only get 2 fractions in, so they got 24 gray or that. And, we've had sort of mixed success, but it's hard when you're dealing with, a number of 1 or 2 animals to extrapolate that and draw any conclusions. So, that's where the basic science sort of research approach sort of kicks in, and that's where, the research funding really helps us answer some of those very basic sort of questions. So the grant that Danny's Foundation has provided general support for is going to fund a project where we look at exactly the question you're asking, which is what is the minimum dose that we can reliably get good tumor control. And so because we have our experience in the canine model, what we're going to use is we're going to use these athymic rats. And so these are rats that don't have a thymus, and the thymus is involved with your T cell production, so with your immune production. And so a lot of the basic science sort of rodent work in both mice and in rats is done in these athymic models, because it allows us to introduce a cancer cell line that isn't a rat or a mouse model. So in this one we're going to be using an athymic rat, and we're using a rat instead of a mouse because of its larger body size. And so for the radiation therapy aspect, that allows us to deliver the sort of doses we need to. So we're going to use a total of 24 rats, and we're going to have 3 different doses. So one at 30 gray, so 3 by 10, one at 11 gray, 3 by 11, and one at the current dose that we use in the dogs at 3 by 12. And so they'll get a total of either 30 gray, 33 gray, or 36 gray. We're also going to have another group where we just irradiate normal bone, because that's one of the other questions we want to look at is, what is the effect of that sort of dose on normal bone, so without any tumor cancer cells there. This is where Anthony comes into play. So Anthony's a PhD student in the School of Biomedical Engineering, and so that's another thing that's not unique to, but a special part about CSU is that we have very good collaborative relationships with our other colleges on campus. So the engineering school that has a biomedical engineering, and obviously they deal in devices, they deal in computational models, they're the brains behind the gadgets that we end up putting into cancer patients. So it sort of crosses biology as well as mechanics and all those sorts of things. So our goal is to sort of figure out, how can we, what's the lowest dose that we can get effective local tumor control, as well as looking at some of those soft tissue effects. I think I covered most of that. The other nice thing is in our lab, so there's a number of ways we can follow the response to therapy, and from an animal welfare perspective, we want to use the minimum number of animals that we can. And so we're actually using a cell line that is transfected, so it has incorporated in its DNA the luciferase gene. And so this is the same stuff that makes fireflies, fluorescent. And so we can take some of that DNA, put it into the cancer cell, and then we have a special camera where we can follow them, and it'll sort of glow up as a fluorescent sort of color. And so that allows us to use the same animal and then sort of follow it, once a week and say, are the tumor cells still growing or not? Whereas before, the only way we could tell that is to, kill the animal and then do histology on it and look at it under a microscope slide. That's a really good part of it. The urine PYD, so when bone is being destroyed, it will release type 1 collagen, which is what's mainly made up in bone, and it'll get released into the bloodstream and then excreted out through the urine. So if you have a lot of bone turnover, so a destructive bone tumor, those levels are going to be high. If the radiation therapy is effective in stopping that bone turnover, we expect to see those levels drop. And then we'll do the sort of standard lameness and acute radiation effects scoring, and then eventually, after we've followed them for a period of 6 weeks after they've had their radiation therapy, the animals will be humanely euthanized, and then we can collect the legs, and so we can look at them histologically, and then in subsequent stages we can do biomechanics and things as well. So this is an imaging plan, so the same as we did the dogs, except this is a little rat, and it's really, really cute. You can sort of see, so this is a rat body here, this is the leg, and so we've injected the tumor cells into the distal femur, so that's a common anatomic location where osteosarcoma or any other bone tumors can occur. And then same as we saw before, we can actually get different beams, and we've done some little pilot studies already, and we found that we can reliably get 36 gray into a little rat bone. And then our histologist is just looking at the first set of pilot animals that we looked at now. So here you can see what we do is we take X-ray pictures or radiographs, and so at the first week, this is at the time of injection, and then we follow them, and then you can start to see some of the very sort of characteristic bone loss appearance that's associated as the tumor grows up until it gets to a very much more advanced stage. But this is after the radiation therapy, so this is actually dead bone. So this is one of the ones that had radiation therapy. This one here, this is this bioluminescence imaging that I spoke about. So with we inject an enzyme that sets it off, and so if there's tumor cells there, we can image it like this. So this is the rat under a general anesthesia. On the first week, you can see a very large intensity of the fluorescence there. Same again the week later. Then we gave the stereotactic radiotherapy, and so diminished it a lot, and then went away for the next 2 weeks, and then we started seeing a little bit of recurrence coming back at the dose that we used in that particular animal. So it allows us to use the one animal throughout the whole study and say tumor is present, then it goes away, and then in this case it came back. So that's how we can say, is the treatment working or not? Some of the things that we can look at, using this sort of rodent model is what are the different fractionation schemes? So are 3 fractions better than 2 fractions better than 1 fraction? Maybe 5 fractions is better, but we can get a better feel for that. Are there other things that we can do? So there's radio sensitization, so can we combine it with chemotherapy? Can we use radioprotectants to sort of try and decrease the dose, still get local tumor control, and so decrease the incidence of side effects, either acute or long-term. Regenerative strategies, and this is the main part of Anthony's PhD project, is using this model, and so trying to get bone to re-heal after it's been irradiated is very, very difficult, and so stem cells are one of the possible ways that we could try and get new bone to grow. So what we're going to do is, after we've irradiated these and shown that we've killed the tumor, is then inject these rats with rat mesenchymal stem cells, and then see if we can get new bone to grow in that irradiated environment. The nice thing with this model is that because it's an athymic rat, we can put any cell line that we want in there. Obviously our primary research interest at the moment, because of our dog experience and our experience with osteosarcoma in people, is an osteosarcoma cell line that we're using. But we could equally use a human Ewing cell line in this athymic rat model, and then look at radiation strategies there as well. So I think that's it in terms of the presentation part. So we're happy to answer any questions. This is just a small snippet of, you know, the wider projects that are ongoing here, and we'll get to go down and see some of those as well. But